Hi, good day. I'm Farzana Diedrichs. I'm a research trainer within the Public Health Alliance for Genomic Epidemiology, also known as PHA4GE. And on today's PHA4GE podcast, we have a very special guest with us, Joshua Levy, all the way from Scripps Research based in California. And fun fact, Scripps Research is held as one of the largest non-profit biomedical research facilities. And with that being said, Josh has also worked closely with us within PHA4GE as a part of our sub-working group within wastewater surveillance. And that is part of a larger network, which is the Bioinformatics and Pipeline and Visualizations Working Group. Yeah. So welcome, Josh. Without further ado, welcome.
Thank you. Yeah,
good to have you here today.
To be here.
We're going to start off, and we're going to dive right into it. We're going to start with a brief overview, or basically, what is your understanding of what is wastewater surveillance? And then, just to touch base also on how has it renewed the importance as an early detection with overall.
Yeah. So wastewater surveillance has been a major focus of our research group over the last few years. Especially has been something that during the COVID-19 pandemic has become a very important way of tracking community-wide spread. So wastewater surveillance leverages existing waste aggregation and transport within communities. And this works from the scale of hundreds to thousands to millions of people. And scales well with the size of...
On a global scale.
At the global scale. And it's very adaptable. So, what might work in Cape Town or in San Diego, where I'm coming from, these methods can be adapted so that they work in any environment, whether that's, wastewater treatment plants where waste is being aggregated from, entire urban areas, all the way to, individual kind of canals or other surface waters that might be contaminated with wastewater.
Yeah.
And the goal here is that we can leverage the fact that during an infection, viruses, bacteria, other pathogens, usually non-viable, but sometimes live versions of those pathogens are shed into the waste streams. And then we can detect them for the whole of the community in a single place. And our group tries to leverage that to do community-wide tracking, not only of pathogens in terms of their overall abundance, but also their evolution and their spread. So that's...
And I think that ties into what possibly would be my next question, is that I know your expertise is so wide. So with working in wastewater surveillance, and I specifically know you've done previous research in San Diego, where you mentioned you're from, how is it, you may have mentioned it, but the techniques and the approaches that you've used in the setting, how does it, should I say that is my first question, and then how would you say that compares to your low resource settings with your work that you've done, possibly Nigeria, for instance, like you've mentioned? So,
So at the end of the day, one thing that's a very, large priority for us is to make sure that we're developing methods that are not just methods for research, that these are methods that are meant for public health surveillance, which means the tools that we use, we try and make them as sensitive, but as also as cost efficient as possible.
Yeah.
So a lot of the sort of approaches that we take for quantification and for sequencing can be done anywhere. And truly, I mean anywhere. I mean, for sequencing a wastewater sample in San Diego or South Africa or Nigeria or Malawi, we can bring the costs down into the order of tens of dollars per sample.
And I mean, that's amazing. That would be the end goal, to accomplish that, in both your high-end countries and your low resource settings.
So it's really it's twofold. So one is this is how you actually get the work done. If things are so expensive, right, it's not sustainable. And that means that the public health labs and institutes within countries can't possibly fund work when a single sample might cost $1,000 US.
Exactly.
When you bring the cost down to $10 to $30, all of a sudden, if you can monitor an entire city of millions for that cost per day, it's quite viable most places in the world. And that means that we're also getting in more and more data to really think about what are the infectious disease problems that our world is dealing with. And that then integrates directly in with governmental and larger watchdog institutions. So then we work with folks like PHA4GE to develop standards, as well as to actually track the spread of these pathogens globally. And the tools that we use actually work quite well, not just for COVID-19, which has been,
And I know that's where you initially started with
Exactly.
with SARS-CoV-2, the virus.
Exactly.
And then expanded from there. So yeah.
Yeah.
The next thing that I probably would like to know is how has the techniques, specifically your next generation sequencing techniques, NGS and Oxford Nanopore Technologies, your ONT, how has that enhanced within the field of wastewater surveillance?
Yeah, so wastewater is a very unique sample type. It's different from a lot of other kind of infectious disease monitoring or surveillance approaches, because if you or I were sick and had a nasal swab done or a blood sample taken, odds are, if there's a virus that's causing this, that we just have one version of it, right? But in wastewater, you've got all of the infections in the whole of the community, all that are being aggregated into a single sample.
Yeah.
And older fashioned sort of Sanger-style sequencing, when you would sequence, you would really just get one sequence. But we know for wastewater that it's this assembly of many, many different sequences, many different viruses or bacteria, all in one sample. So what NGS allows us to do is, instead of just getting one sequence, which we already know wouldn't be representative, instead it's an assembly of sequences from across the community that we can then basically pull out the individual pieces and say, actually, these particular genomic components come from this particular virus variant, and these ones come from another one, and those two different viruses might have different implications for public health.
And that is exactly the aim, to get more efficient with the checking of the wastewater surveillance. So you and your lab, you've developed a tool called Freya. And tell us more about that. What does the tool do? What is it able to track? How is it able to track and disease and monitoring the outbreak?
Yeah, so Freya, as you brought up earlier, was part of a project that we had started originally just in San Diego. We needed a way to take this next generation sequencing data and convert that into meaningful estimates of the different virus variants and their associated prevalence in the community. But no method existed, so we leveraged the information that we had at our hands at the time, which was, one, that we knew that there was all of this clinical sequencing that was being done, where individual sequences and virus variants were being identified and brought into a single sort of database. And we knew that if we had access to that information and the associated definitions for all of those variants, that we could build a mathematical method. My background is actually in applied mathematics.
I know, quite awesome.
And so then from there we were trying to think about how could we take that information, the actual sequencing data, and a mathematical sort of formulation to pull this all into something that we could use from a public health standpoint. What are the changes in those variants over time? And then we can monitor this from, in San Diego, we were doing this from a wastewater treatment plant from about 2.3 million people at this wastewater plant. And it worked surprisingly well. And we showed our original results. And then we started to think about, we work in a lot of places in the world where what we have in San Diego doesn't necessarily make sense in terms of the sampling and all of those sorts of approaches. So we started working with colleagues in Malawi, as you mentioned, Nigeria, here in South Africa as well, and in many other countries have started to use Freya as a public health tool to see if these sorts of methods would work elsewhere. And we found out that the sampling approaches need to be modified differently. You need to sometimes take different variables about the local environment into account, but you're able to do it. And if you're using kind of these low-cost approaches, it can work quite well. And then more recently, we've generalized this sort of FREYA approach. So now we are starting to do this for a host of different infectious diseases and pathogens that are
and I think I mentioned that before, because I know you mentioned with FREYA, you started on SARS-CoV-2, but then you expanded and worked with monkeypox, influenza, and so forth.
Exactly,
But bringing it more into context, the tracking as per se, I know you've briefly mentioned dashboards and so forth. So how does that work?
Yeah, so there's a few different sides of the problem here. One is, how do we actually make sense of the data in the first place? That's a very important question, and being able to get that information to public health stakeholders, public health officials, members of health ministries, that's very important. But we also need to have ways to get that information to the people as fast as possible and in a way that people can understand.
Exactly.
So it's been a major focus of what I've done, especially working with the NICD here in South Africa, is to think about how do we take this information, which is complex genomic data, and convert that into something that is readily user-friendly and intelligible, that if you have it, you load it on your phone, you can see, oh, there's this new virus variant around, and you can read about.
And exactly that.
means.
If you look at SARS-CoV-2, the different mutations within SARS-CoV-2, you're able to track that and see it.
Yeah.
Cool. I think that's a lot for the tools and the bioinformatics part, but I'm going to bring it more into context with a previous interview that you had once. And you mentioned ethical considerations around wastewater surveillance. And you called specifically wastewater a unique modality for surveillance. Would you mind maybe just elaborating on that?
Yeah.
What your thoughts around that?
Sure, yeah. So wastewater is a pretty unique surveillance modality. It, in many ways, allows us to not have to worry too much about many of the core kind of public or personal health identification, identifiability concerns that you have with clinical samples. Because the samples are fundamentally kind of anonymous. You take one sample that's representative of a whole community, you have no idea where that virus came from, who possibly could have actually been infected and then led to the detection that you have in the wastewater. So that doesn't mean that wastewater does not have any ethical concerns. The scale at which you choose to do it and how you choose to report that information does really potentially have some significant social and actual stigmatization related concerns. So, for example, you brought up Mpox surveillance, which is something that we've been working on quite a bit. Mpox is a pathogen that affects often stigmatized populations. And if you are doing wastewater surveillance that is focused specifically on a neighborhood that could be targeted, or a group of people within that neighborhood that could be targeted for the information being present, you have to be very careful about how that information is handled. It's not to say that the work shouldn't be done, but it needs to be, that information needs to be brought to the public in a very careful way. So those sorts of concerns are very real.
Of course.
There's also these larger questions of what do people gain from this? Does this, just because the work is done, does it mean that there is actually what's called beneficence or kind of improvement of the living conditions and the actual health of the community from the work there? If it's not actionable, if we can't get this information to people in public health so that they can start vaccination campaigns or get the medicines to the communities that need them, we have to really think about why we're doing the work in the first place. That's not to say that we shouldn't do it, but it's starting to think about, we can now operate at this large community scale. How do we get, an actual positive step in the right direction in terms of
And I think that
health.
sort of hinders the process sometimes, because ethics always plays a major factor when you're working with anything for that matter. So, yeah. I think we'll end it off with one last question.
Sure.
If you could just give us a brief introduction as to how your current work within Scripps and how does it align with PHA4GE, if at all? And then potentially, is it still, in fact, contributing to wastewater surveillance? And on what scale? Is it at a global rate?
Sure. Yeah, so the work that we do at Scripps, and I'm a member of the Anderson Lab at Scripps Research, so I only can really speak to the specific part that I am kind of leading there. But that work is focused on the development of basic tools for wastewater surveillance. There are some members of our lab who are also doing development of the sequencing-related tools and basically the quantification tools as well. So we're trying to basically develop the kind of methods that can be used for the field to some extent. We're one of many that are doing this. This all then connects into PHA4GE's larger kind of standards development and best practices development, especially within the wastewater surveillance field. And what we're doing basically is, you mentioned Freya and related packages that we've been part of developing. We're trying to figure out what are the best practices for the field. How do you get from sample collection, well, even before sample collection, how do you choose the sites that you wish to sample from? Then how do you collect those samples, process them in the lab, and then take that from analyzing the sequencing data all the way to dissemination. So we're trying to provide information on the sort of like standard protocols without an overwhelming amount of data, but in a way that allows people to get started if they haven't yet done it. So no one's starting from total ground zero. You can build off of the best practices that PHA4GE has sort of aggregated.
And with that, I think like when you say people, you imply not just us as bioinformaticians, but public health officials, your environmental scientists, those that does not have that bioinformatics background to apply that knowledge and sort of do the same thing,
Yeah, and that's a huge focus of what we do is, certainly speaking about Freya, which has been probably the most commonly used tool that I've been involved in developing, we see that, while traditionally these tools were operated and used mostly by trained bioinformaticians, in fact, that's not always the case. That we've now been able to work with folks like PHA4GE, Staff B in the US, which is the state public health bioinformaticians group, as well as other outside groups like Theogen, to build public health ready packaging. So basically, even if somebody doesn't have bioinformatics background, we can allow them to use the tools in this way. And also one amazing thing that PHA4GE does as well is their PHA4GE, I do my small part in the larger PHA4GE world, but is that building that the younger generations and teaching people the skills so that you have people operating at all of these different levels, from no bioinformatics experience up to experts, and establishing those systems globally.
An all-rounder. But thank you so much, Josh. That's about it. It was good to have you.
Awesome. Yeah. Thank you very much. Glad to be here.